Okay, welcome back. We left our last lecture with fractionation, and so we have proteins and organelles in each fraction. How much do we have? Okay, how much proteins do we have? Every organelle in the cell has specific organelle proteins that are unique to that organelle. There are enzymes in each organelle as well that do the business of that organelle, which we will get into. Okay, enzymes. So the concept, and this is before we get into spectrophotometry, is that we are interested in total protein and how much of that total protein is in that fraction is the Golgi nucleus, lysosome, plasma membrane, mitochondria, specific proteins and enzymes. In cell biology, we call those markers or marker enzymes or marker proteins, so marker. So we know if we studied each fraction in our fractionation, okay, markers, protein or enzyme, okay, protein or enzyme markers. So we can get an estimation of which fractions contain which markers. So we can figure out, hey, there's a lot of Golgi marker in these fractions. You would predict there are Golgi structures, structural Golgi membranes there as well. So when we're breaking a cell open, and if we want to study the Golgi, that's how we would narrow our search of the fractions. So if you remember the fractionation from the last lecture, okay, a way to measure the amount of proteins in a sample, and this is a biochemistry approach that I know you will need to know, is spectrophotometry, and it's a technique that uses the absorbance of light by your sample depending on the wavelength, and that absorbance correlates to the amount of protein. You can use UV or visible light, okay, and it measures, like, the amount of light absorbed or transmitted. So as you think about a muddy river, a clear river, if you shine a flashlight in a clear river, you can pretty much illuminate the bottom. There's not a lot of light being absorbed, okay, in a clear water. There's nothing there. In a muddy river, the light is absorbed by all the mud and the dirt and the thing in the river, and you can't see anything. So there's a high level of absorption versus a low level in the clear river. So think about that as a concept. So in our fractions, then, there are fractions that have a lot of protein. There are fractions that may not have a lot of protein. Okay, when we purify from the cell organelles, it would be to our advantage to limit what we're studying. Okay, just focus exactly on the fractions that have proteins in them, which probably means they have organelles in them. Okay, and so the absorption versus transmitted, you can actually get both information, and we'll see how that plays in just a second. This is the cartoon diagram of a spec, and there's a light source. That light source can be UV, or it can be visible light, and it just depends on what you're doing and what you're trying to detect. The light is released and it's concentrated, and so, by the way, if we look at the spec machine here, this little blue area is where we drop in a sample in a glass or quartz cuvette. A cuvette is a tube filled with solution. Okay, glass is much cheap, quartz very expensive, and so the light then is coming from a direction that is going through the sample some which way. Okay, I don't know the direction, but you pop this open, you drop your sample in, so the light's coming through, and so you get the intensity of light originally, the IO here, and then there is a little aperture that you can change the shape or the length of the aperture. Light is focused in through there, in through your sample, and then the light that comes through then can be measured. Okay, and so it's kind of hard to tell, but you see that it's dark here and it's coming through, it's lighter, the wavelength, because there's been some absorbance. Okay, and then the signal is amplified and you get a reading. Okay, this is the output, 0.26. So unless you do a standard curve, which we will talk about, you won't know what that reading 0.26 really, standard curve, really means. Okay, so you have to know the standard curve, and this would be where you would see your reading here at this point. Okay, so this is another cartoon. This is showing the IO, and this is showing the I, and this is our sample comes through. So you can measure transmittance. Transmission percentage is this amount of light divided by the original amount of light. So that tells you percent transmit, in other words, how much light came through. Absorbance has an equation. It's the analog of the transmittant. Okay, it's inversely proportional. It's the complement of it. It's called absorbance, and the amount of light that's, so if you think about that, this is what came through. So there's some light here that was blocked and absorbed by the sample, and it did not come through. So these machines can, there's a little switch, and you can get transmittance or you can get absorbance, your choice. Everything is based on absorbance from all the tests we do here, but you can figure out transmission. Okay, so at UV wavelengths you can get nucleic acids or proteins. Here's 260 nanometers or 280. So protein is at 280, nucleic acids 260, and you can get the absorbance here. Okay, and you can actually look at the ratio of the two. So you can monitor proteins in each fraction. You can use UV light, 280. The issue with UV light is it only detects aromatics R groups. Okay, R groups, and those are tyrosine, tryptophan. So you can get some bias if a protein doesn't have a tyrosine or a tryptophan. You would not get much absorbance. If a protein has a lot, you might get more than you would predict by the amount of protein. So I'm kind of showing you that this is usually not what we do. It is a quick way. If you have this, doesn't need any chemicals or any kind of reactions. Once you have a UV spec and the UV cuvettes, that's your investment. You don't have to buy any kind of chemicals. You've got it forever and ever. But there are some issues. So again, we're looking at DNA. Here's just DNA. You can get the bases GATC here, about 260. And you can also figure out double-stranded and single-stranded DNA as well if you're doing. And again, this is our story with amino acids, aromatics here. Basically tryptophan and tyrosine. So DNA 260, protein is 280. The absorbance and the comparison of 260 to 280 will measure purity of DNA. Remember, there are histones. Histones are proteins. Okay, histones, proteins, and they wrap the DNA. So the amount of DNA is wrapped by histones, and the amount of protein then can be predicted because they know the structure of DNA, and so they know the amount of histones per turn of the DNA. And the reading should be 1:7 to 2. In molecular, we can go into this, but I'm just kind of introducing it here. And so the absorbance should be, it's about 1:7 to 2 greater than the amount of protein. So just the proportion of DNA to histones there is about 2 to 1. Okay. So other methods, again, we got to use the UV light. We can figure out the amount in a sample. In other words, how much DNA is in this sample. There is a known value known as an optical density, which is an absorbance value, is equal to 50 nanograms per milliliter of solution of double-stranded DNA is 1. So if you had 2, it would be 100. If you had 0.5, it would be 25. So there is some calculations that you can figure out pretty quickly how much DNA is in a sample. Okay, and so for there is a linear relationship between absorbance and concentration, especially in proteins. And there is a chemistry that uses copper ions to detect proteins. And so if we're given a known protein and we do at different known concentrations, and the little squiggles are the concentrations, we can plot the absorbance versus the amount of color. The copper is a bluish color, kind of a light blue. And then we can detect unknown. Here's an unknown. And an unknown would be about, in this case, 0.125 maybe. And that equals to about 0.8 absorbance. And we know how much protein is here. And so we know here. And so we can calculate the amount of protein at that point. This is another standard curve. There's another reagent known as BCA reagent. Here's the absorption at 280. So BCA is visible light, and we're going to touch on why visible is the best. But this is UV. Okay, UV. And so they're comparing known bovine serum albumin. So this is from cows. Inside their serum, there's a major protein called the albumin protein. So you can get a lot of that quickly. And so this is the optical density on the Y, and the X is the amount of BSA mg per mil. And so they do the concentration here, and they plot the different readings. Here they are, and then the amount of BSA at that point. Okay, so when you have an unknown, then like what I just drew, your concentration is somewhere 1.4 maybe. Okay, so this is the unknown. Okay, so if you just got an optical density of 0.07, what does that mean? I don't know. You don't know until you've done a standard curve. Now you can find 0.07, draw it to here, and drop down. Certainly there is math equations to describe this, and you can use the equation to predict the amount. So bioinformatics has software that usually comes with these spectrophotometers that you can use. You can just calculate whatever absorbance, and then it will tell you how much protein is there. And this is UV light, and it's a little different because this is UV light estimates protein. Well, this is here. This is here. So clearly different. So it's not as accurate, but gives you an aspect. So estimating protein concentrations using UV light, again, things like phenylalanine, tryptophan, tyrosine. You don't need any kind of chemistry, no kind of colored reagents, no kind of kit. You've got a UV and a quartz cuvette. You don't chew up the sample. You can reuse the sample. Absorption does give you concentration. Problem is there's contamination. And most specs are used in UV and visible. And again, you can do UV at 280, but it's not accurate, or not very accurate, because the amount of aromatic amino acids in proteins varies. And that can be an issue. Also, if there's any DNA, that can contaminate. So there's a contamination accuracy issue using UV light, but it's quick and fast. So pros and cons. So most researchers, if they fractionated a cell, will probably use UV light. Visible light. If it's the first time they're doing it and they're not going to repeat it, okay, they can use UV because it's quick. But if it's something that's going to be published, they probably will use visible light. In visible, we have to have an indicator. It's usually a color metric. The most widely used is called Bradford. Doctor named Bradford invented it, and he used it to figure out unknown concentrations of proteins. Because in visible light, if you looked at proteins, they're colorless, maybe a little hazy, the colors. So proteins are mixed with an indicator, and it makes them visible in the in the visible light. So remember our protein structure: the central carbon, the N-terminus of a protein, the C-terminus of the protein, the R group. Okay, so if you hadn't memorized this, might as well go ahead. If you do biochemistry, you'll have to memorize all the structures. That's coming. If you go to medical school, you have to memorize all the pathways synthesizing amino acids and breaking them down, and the diseases associated. So here you go. Start chopping the wood now. They are linked by peptide bonds. There are these amino acids. There is an R group, but it does vary. There's 20 common amino acids, 20. And the R group will determine how the indicator interacts. So there are Biuret and Lowry. Biuret's not very good when concentrations are low. Lowry is very sensitive to low, but not good with high. So those are not as common as the Bradford. Bradford is also known as dye binding. Kamasi Brilliant Blue G20 250, and you monitor the color change. The dye binds to basic and aromatic amino acids, arginine especially. The reagent, when it comes from the kit, is a brownish color when you mix with a sample, and over time you let the chemistry, it turns blue, and the amount of blue color determines the amount of protein in the sample. So the degree of color is dependent on the number of protein molecules. You can use the visible light with the Bradford. Darker the blue color, the higher the protein concentration. So again, why are we trying to figure out the protein? When you fractionate cells, again, you don't know exactly where all the organelles have landed or all the proteins you might be interested in studying are. So if you have 100 fractions, it's best to figure out how much total proteins in each fraction. If there's not a lot of sample there, that's probably not a fraction you need to use and investigate. There's a delay aspect. Things can degrade. I know we used protease cocktail, but that's no guarantee and could fail. So you need to be rapid, you need a reliable, and you need not to have any contamination along the way for your assays. So the time when accurate proteins are needed, after you've done a fractionation step, you want to know what's in the protein. Did you get rid of any unwanted proteins? We'll talk about protein fractionation more, and you'll see when we do chromatography, we could this step comes in. So just keep that in the back of your mind. Specific activity. Every enzyme has specific activity. It's the amount of enzyme by its activity. 100% is pure enzyme, right? So if we have marker enzymes for Golgi, what's the specific activity for that enzyme in the fraction? If it's pretty high, that probably is a good indicator we have a Golgi organelles there. Okay, if it's not so high, then there's probably a lot of other organelles or other things there. Okay, so that gives us a clue about our fraction. So again, it's the Bradford is what we're doing through. It's just talking about the instruments you need and sensitivity. Obviously, we want enough cells so we can detect proteins. So if you're, and again, this is all empirical. When you break cells open and you fractionate, it's empirical. Unless, you're doing exactly what somebody else has done before, most likely you won't be. So it's empirical. So you will need to know right off the bat when you get your, say, if you're doing differential, your first pellet and soup, you can do protein analysis on both. You should get a nice number it's like, yes, I have plenty of protein here, versus I can't detect much protein. Well, maybe you didn't start with enough cells. So there are figures. People say how much protein on average per cell. So if you figure out how much protein, you probably know how many cells are there. So Bradford's the most widely used. It uses the Kamasi Brilliant Blue, and it binds to the basic and aromatics. And you go from a brownish, that's detected at 465 and you switch the absorbance to 595, that's where you're going to catch the blue. Okay, and you get an optical density, a number that you compare fitted. When it says fitted, that means compared to a standard curve, and therefore the protein is determined by the fitting there. 3 to 5 kilodaltons, that means 3,000 to 5,000 daltons. Each amino acid on average is 330 daltons. So this is something you always remember now. So if your protein is 33,000, okay, that means it's 10 amino acids, okay, which is a very small protein. So I took the molecular weight, divided it, right, by 330, and that gives you the amino acids. Okay, in molecular biology, it's really important if we're hunting for genes. It's good to know, if we start with a protein, how many amino acids are there, because remember, every 3 amino acids, right, is 3 bases. Okay, 3 bases. So that means this gene is at least 30 bases. Okay, so it's a small gene, small protein. So it's a colorimetric reaction from the brown to the blue, and compared to the standard curve concept. Here are the standard curves, standard procedure, standard curves, the amount of protein on the X and the absorbance, the optical density on the Y. And it talks about, here's a concentration of 60 micrograms per mil. If you diluted your sample 1 to 10, then it's really 6,000 micrograms per mil, which is the same as 6 milligrams per mil. 100 mils of a 1 to 10 dilution gives a comparable reading to 60 micrograms on a standard curve. These kind of calculations here, okay, are not a big deal in cell. You probably did some of this in micro. I know you're going to do a lot of it in the chemistry, so I'm assuming you've gotten and can figure this out if you need to. Here's the brown to the blue. Okay, a lot of protein here, not a lot there, if any. Here is a standard curve of micrograms per mil of protein. Notice very blue, a lot of protein. Light blue, if mainly not brown, very little protein. Here's some of our fraction samples. So this is pretty good levels here. This is a good level. You see what I'm focused on where I see a lot of the proteins here. So these would be key fractions to study, okay, when you're fractionating a cell. They have plate readers that send a beam of light through each hole and you get a reading. So I showed you the cuvette, that's one sample. Now we got 96 well plates, 382 well plates, and I think they've got even smaller. Obviously the amount of sample goes down with the plate, the number of samples, right? So this will take it inside and shoot a beam of light in every little hole, and you will get a reading that will be saved. Optical density. Here's your standard curve. You take known protein samples, concentrations, and measure the absorbance and plot it. Okay, here it is. And looks like somebody's selling something that makes a better standard curve. Okay, here's the standard assay. So you're getting protein standard, bovine serum albumin versus absorbance, and they're showing and plotting. And this is, here's a blank, so it's a negative control. And here's where you have a lot of protein here. So you need a negative control with no protein. A positive control has protein. In other words, if you don't do that, then if they all are brown, you can come out, oh, there's no protein here. What's your positive control? Oh, I didn't do one. Maybe you forgot to add your reagents in the Bradford. And so maybe you forgot to add Kamasi blue, so it just stayed brown. Okay, so this is Lowry, and it just shows the not very sensitive compared to the Bradford. Here you get some readings. So Bradford is what we're using, and that's how we look at our protein fractions here. So you extrapolate from the standard curve and figure out the unknown here, and they're just showing this again. So if you had an unknown, right, that came here about 0.7, you can drop down, and it's about 12,000 microgram per mil, or 1.2 mg per mil. And yes, you need to be able to jump from nanograms, micrograms, milligrams, grams. Yes, if you're going to be a doctor or dentist or pharmacist or something, yes, you've got to know it. So review that if you don't know the units. So first time we do a fraction, we could use UV light. Gives us a quick and dirty view of how much protein is there. We see 4 peaks, and here's the fraction numbers. So we could collect these fractions for further study. If we wanted to study for marker enzymes, okay, we would study. So this probably, you know, this could be the Golgi, this could be the ER, whatever it is, okay. And so we're able to separate these in the fraction. The only reason I want to show you this was to remind you that we can use facts here. And looks like this lab or whatever was breaking down adipose tissue. There's adipocytes, apparently there's stem cells, these ASCs, there's macrophages. And so they have they chew up the collagen that holds the tissue together, and they centrifuge and then plate, and they get the floating cells, and it looks like these, maybe these stem cells are the pellet, and then here they are attached, and these are stem cells, so they can turn into, they have multi potency, so they can turn into things. And it looks like under stress conditions you can get a cell pellet, Muse cells, you plate them, and you get these clusters of Muse cells. I don't know what that term is. First time I've ever seen it. Looks like some type of cell structure when there's a stress condition. Never heard of it. But anyway, this is reminding you of the 4 quadrants of fluorescence activated. So PEA, which is also, I guess, CD105 production on the cell, and SSEA3 production. So it's green. That's probably, yeah, phytourethrin is red. So apparently what they're finding is most of them have a red and green. They're in this quadrant here. You can always look at invisible light to see the structure of what you fractionated. So just to let you know, here's these floating clusters, here are these aggregates. So one can obviously do protein levels, you can do facts, but then you can also take the samples and look for structure there a little bit. So that's kind of really interesting. So I was trying to tie in where we've been to fractionation, to looking at cells, to measuring different cell types and protein levels. Here's kind of putting it all together. Okay, well, that's my 10-cent tour of figuring out protein amounts. We will use this again in protein fractionation. Okay, we will pick it up with cell-free systems coming to your theater very soon. Thanks for listening. See you in class.